We have Dr. Meira Epplein from Duke University. She's an associate professor in population health and health sciences and medicine, and she's also a member of the Duke Cancer Institute. She's a cancer epidemiologist who's interested in modifiable risk factors in underserved populations with a focus on infection and cancer. Meira has been very involved in TRISEM. That includes attending and giving talks and participating in our Evolutionary Medicine Summer Institute, including a panel we held on health disparities. So thank you for everything you've been doing within TRISEM, Meira. We really appreciate that. And she has with her a co-author, Dr. Julia Butt, who's a senior research associate at the Infectious and Cancer Epidemiology Research Group at the German Cancer Research Center. So thank you very much for joining us as well, Julia. They shared a just published paper on with the title Differences in Antibody Levels to H. pylori Virulence Factors VacA and CagA among African Americans and whites in the Southeast US. They'll give about 20 minutes, 15 minutes of presentation related to that, and then we're going to open it up for discussion. Okay, take it away. I think we're ready to go.
Great. Thank you so much. And as I share my screen, thanks to everyone for joining. Julia and I are epidemiologists. Julia also has a cancer biology background, but we love talking to you guys. We love your input and your perspective as evolutionary scientists or folks interested in evolutionary medicine. So we are completely open and excited to hear your thoughts on our area of research. So I wanted to start with just the big picture about why Julia and I are so passionate about this area. And that is, in general, I think people often think of cancer as a chronic disease, which of course it is, but they don't realize that a significant number of cancers are actually caused by infection. So currently it's estimated about 15% of cancers worldwide are due to infection. And that's about 2 million cancers a year arising because of infection. And the infections that are causing these cancers, so some of them are very well known to you. So there's HPV in blue, human papillomavirus, for which we have a vaccine, and we're making great strides in reducing cervical cancer, hopefully head and neck and anal, a number of the HPV-associated cancers. In yellow, we have the hepatitis B and C viruses that cause liver cancer, and we have a vaccine for hepatitis B virus, and we treat hepatitis C virus. The other, the purple slice, includes Epstein-Barr virus, liver flukes, schisto. But look, the largest, the lo and behold largest, the infection causing the most cancers is Helicobacter pylori, one for which we have no systematic treatment or screening program, at least in the United States. And yet H. pylori is associated with over 800,000 gastric cancers a year. So what is Helicobacter pylori? It's really fascinating bacteria. It's gram-negative. It's a spiral microaerophilic bacterium, which means it requires oxygen, but not as much oxygen as in the atmosphere. We still think it infects about half the world's population has chronic H. pylori. So obviously, it's living in our stomachs, and for the most part, not doing much, and you don't realize it's there. It's highly genetically diverse. So we believe it's been colonizing the stomachs of humans for over 100,000 years. And I'll get to that more in a minute. The mode of transmission, we say it's uncertain. We know it's oral, fecal oral, vomitous oral. We currently say that humans are the only known reservoirs. Certainly people look for Helicobacter pylori in water. I think none of that is substantiated yet. Very excitingly, there's effective antibiotic therapy. So it used to be you only needed one antibiotic and you could get rid of someone's, excuse me, H. pylori. We're now up to triple therapy, which is 2 antibiotics plus a proton pump inhibitor. And unfortunately, standard triple therapy isn't working as the bugs are increasing their antibiotic resistance, and often we're using quadruple therapy now. Nonetheless, it's a 14-day regimen with usually 2 antibiotics that are available in generics, plus a proton pump inhibitor, and sometimes plus bismuth. But it is a feasible treatment that can be very effective in curing infection. Of course, we have the problem of increasing antibiotic resistance, and currently about across the board, we see when people are given antibiotic therapy, we have about a 70% cure rate. So that's not great. Back in 1994, IARC, the International Agency for Research on Cancer, declared H. pylori a class 1 human carcinogen. basically primarily on its relationship with gastric adenocarcinoma. It also causes other gastric diseases. Speaking of which, you might know these two guys. So H. pylori became really famous in the 90s when these guys, Barry Marshall and Robin Warren, discovered H. pylori and were later awarded the Nobel Prize. And they were awarded the Nobel Prize for their discovery of H. pylori's association with ulcers and gastritis, so not yet to gastric cancer, but because people didn't think a bacteria could live in the highly acidic environment of the stomach. So it was really amazing discovery by the younger fellow on the left, Barry Marshall, who actually drank some H. pylori, self-experimentation, no IRB approval, quickly developed severe gastritis, actually faster than he thought, and again, realized that there was a bacterial cause of ulcers, which people previously thought was primarily stress-induced. So the natural history of infection of H. pylori has a lot to do with the age of infection, which is the x-axis, as well as the acid production in the stomach, which is the y-axis. So again, for people with chronic H. pylori infection, they're usually infected as a child, either by their mother or their older siblings, or when they start school by age 5. And you can see that middle bar, so the majority of people who have a chronic gastritis due to H. pylori, it's asymptomatic. So 85% of people, half of you or 30% of you are walking around with asymptomatic H. pylori. The bug is doing its business, you're doing yours, no one's the wiser. It can also lead to duodenal ulcers and to gastric ulcers. And in a small percentage of people, it leads to gastric cancer. But again, that's only about 3%, 1 to 3% of people with chronic H. pylori infection will actually develop gastric cancer, even though it is the leading cause of gastric cancer. So speaking of gastric cancer, the map is really interesting. So in the cancer epidemiology world, we often talk of cancers being developing world cancers or developed head world cancers. And gastric cancer isn't like that. It's across countries. So the darkest blue, these are age standardized incidence rates for both sexes. And the darkest blue are the highest incidence rates and really very high in East Asia. So in Korea and Japan, very high rates, as well as China. You can see we also have high incidence of gastric cancer in Eastern Europe and in parts of South and Central America. Really interestingly, for the most part, Africa and India have low rates of gastric cancer, and we call that the African enigma and the South Asian enigma, because they have a lot of H. pylori. But so far, and this might be changing, we haven't been seeing a lot of gastric cancer. One of the theories being the heterogeneity of the bacteria, which we'll get into in a minute, but also the cofactors. So we know that smoking and high salt intake increase the risk of gastric cancer as well, maybe synergistically with H. pylori. And Western lifestyle seems to also increase risk. So here's a chronological reconstruction for you of the major population events occurring after initial acquisition of H. pylori. So the idea is that the human ancestor acquired H. pylori there with a 1, the black 1, in Africa. And then as people emigrated, so did the bacteria. And one of the really interesting things is we look at phylogeographic origin of bacteria to genotype a person's strain. And actually, it was a great article a number of years ago, I think in Scientific American. It's like, are you your ancestry or are you your bug's ancestry, right? So you could have a match or mismatch. You could be of European descent, but be carrying an HP East Asian strain. So as people emigrated out of Africa, you can see these various lines going on. If you follow the black line all the way to the gray up to number 6, we're going into East Asia there. So we know the East Asian strains are the most carcinogenic, which again follows along with the highest rates of gastric cancer in East Asia. They're also the least heterogeneous. The genetically most diverse strains are in Africa, and it seems that as one moves out of Africa to farther regions, the strains become more homogeneous and more carcinogenic. So Europe is in the green. The European strains are considered kind of medium carcinogenic compared to low African strains and the high carcinogenicity of the East Asian strains. The big red 10 is a really interesting hybridization of the Central, Southwestern Asian strains along with the Northeast African and Middle Eastern strains that then became what we call HP Europe. So as you notice in the big map, the United States is not a dark color. It's not a high incident country for gastric cancer. And yet we have this really big disparity in gastric cancer incidence. So we know in the US, people of color are 2 times more likely to be diagnosed with stomach cancer than whites. So for non-Hispanic whites, we see about 5 per 100,000, and you can see non-Hispanic blacks and Asian Pacific Islanders, it's more like 10. We also see high incidence in American Indians and Alaska Natives. Interestingly, because it's a highly fatal cancer, so I forgot to mention that on the other slide, but unfortunately, stomach cancer is often caught late stage, so it's about a 30% 5-year survival rate. So there's a high fatality, and it actually, well, often when we think about disparities in cancer, we think about prostate cancer, Black men dying at higher rates, more aggressive prostate cancer, but actually stomach cancer accounts for the greatest disparity in death rates. So these ratios of 2.5 and 2.3 for Black men and Black women compared to white men and white women for dying from stomach cancer. And again, stomach cancer is still not a common cancer in the United States, so less than 1% of Americans will develop stomach cancer in their lifetime. We're still talking about 27,000 people and over 10,000 deaths each year from this potentially preventable disease. So people of color are also significantly more likely to harbor H. pylori. And an interesting study came out recently from the Houston VA, where they looked at medical records of everybody who had an H. pylori diagnosis. And compared to H. pylori positive white people, H. pylori positive Black and Asian people were over 2 times more likely to get stomach cancer, even though they all had a diagnosis of H. pylori. Also excitingly in their data, when they could find evidence that the person was retested and had successfully eradicated their bacteria, there was a reduction in risk of developing gastric cancer. So I keep wondering, why aren't we doing anything about this? This seems like a place where we could really jump in and make a difference. And one of the reasons I think is that people feel like H. pylori is disappearing in the US. And maybe you guys have read this a few years ago. Marty Blazer from NYU came out with a book that made like the New York Times, I think, bestseller list called The Disappearing Microbiome. And that we are losing some ancient microbes. And certainly H. pylori is an ancient microbe. And if it's disappearing, then we're also seeing gastric cancer declining, which is true. But the work we've done, and this was just published online in a subset of a bunch of population-based cohorts around the United States, we found that, yes, that by birth year, which is the x-axis, we do see a decline in proportion H. pylori positive for whites, but actually we're not seeing that in our data for African Americans. So not only do African Americans have a lot of H. pylori, it doesn't seem to be disappearing like it is for whites. And in fact, that's increasing the disparity over time. So this is gonna lead to Julia describing more what we sought to explore.
Yes, thank you. So like Meira introduced before, it is not only the case that African Americans in the US are more likely to have H. pylori, but they are also more likely to have gastric cancer. So is it possible that this association may be related to the respective H. pylori strain that is infecting these individuals? And as she said before, H. pylori strains vary in their carcinogenicity, and this relates, among other proteins, to 2 main virulence factors of the bacterium called the vacuolating cytotoxin A, short VacA, and the cytotoxin-associated gene A, called CagA. Both proteins are secreted by the bacterium. VacA then has different effects on the host cell, like vacuolation, like the name of the protein says, but it also has immunomodulatory effects, for example. CagA is directly injected from the bacterium into the host cell by a type 4 secretion system. And it interferes with the host cell cycle, but also with tight junctions or other effects on the cells. Oh, wait, I'm not. Sorry, can you go back? Thank you. So the interesting thing about these 2 proteins is also that VacA, or almost all H. pylori strains, harbor a gene that encodes for VacA, but there are different variants of the gene that determine how much of VacA is produced and which isoform. And CagA is a different story. It's expressed from a pathogenicity island that is not present in every H. pylori strain. So they may vary whether they have this pathogenicity island or not. Now next slide, please. Okay, so to look a bit more in detail on not just H. pylori seropositivity overall, but to see whether a person might be infected with one or the other strain. We developed a method in our lab here in Heidelberg that is called multiplex serology. This is a fluorescent bead-based assay where we have an array of beads with different internal fluorescent colors that can be linked to a recombinantly expressed protein. And then these different bead sets coupled to different antigens can be mixed and incubated with serum. And this Luminex flow cytometer then can distinguish between the bead type and the bound antigen, as well as quantify the amount of bound serum antibody. And for H. pylori specifically, we have an array of 13 proteins so far, including the proteins VacA and CagA that I just mentioned. And we define being overall H. pylori seropositive to being positive to at least 4 out of the 13 proteins. But we can also look into more detail whether somebody has antibodies to a specific protein like VacA or CagA, for example. Next and next. Okay, and this is exactly what we did in a study called the Southern Community Cohort Study, which is a study that recruited primarily low-income individuals in the southern US to assess health disparities in the US. And so we assessed the antibody response to these H. pylori proteins in this cohort. Can you click next, please? Yeah, thank you. And the population we analyzed consisted of African Americans and whites, and we compared the seroprevalence in these two groups. And we found that overall seroprevalence already varied by being African American or white, like Meira showed earlier. With in this population, 89% of African Americans being seropositive to H. pylori compared to 69% of whites. When we look at the virulence factors separately, it's very interesting to see that almost all H. pylori seropositives also have antibodies to VacA. But again, this differs by being African American or white. And then for CagA, in the two middle bars, you can see that not all H. pylori positives are also seropositive to CagA, which kind of reflects that not every strain has the gene to express this protein. And interestingly, the seroprevalence of CagA was more than twice as high in African Americans than whites. Next. So since this is a quantitative assay, we can not only assess whether somebody is positive or negative, but we can also look at the antibody level. And we were wondering whether this might differ as well. So the strength of the antibody response that a person develops to the bacterium. And we again looked and compared the antibody level among seropositive whites and African Americans and found that indeed African Americans were not only more likely to be seropositive to the two proteins, but they also had higher antibody responses, so a more pronounced antibody response to the infection. And if we express this in odds ratios, which we like to do, is that we divided our antibody levels into quartiles and assessed whether African Americans are more likely to be in the higher quartiles than whites. And indeed they are. So in this red boxes, you can see that African Americans are around 6 times more likely to be in quartile 3 or 4 to H. pylori VacA. And below is the same plot for CagA, and here it's 2 to 4 times more likely of having high antibody levels. And because one could say, well, this could also relate to other factors than just the antibody response, like, for example, the sex distribution, or whether more African Americans smoke than whites, or the age of the participants. We adjusted for factors that might affect the immune response, but also with adjustment, these odds ratios didn't change. So this is all interesting to know, but it's just an observations, and we so far don't really know what it means that the antibody response is higher in African Americans than in whites. And there are different options, and I guess the ones that are named here are only a few, is that this dose response association may be an indicator of an active infection compared to a past infection. And what we did was to compare the antibody level in individuals that were tested by a breath test for an active H. pylori infection. And indeed, we saw that the antibody levels were higher in those breath test positive individuals here in gray compared to those that are negative here in blue. And if we apply a cutoff, VacA antibody levels alone show a very high sensitivity and specificity for current infection. But of course, this might not be the end of the story. It might also be the case that the antibody level reflects a more pronounced infection or inflammation that is caused by the host. So in summary, the bacterium H. pylori is the leading cause of gastric cancer. And it's well known that African Americans and other non-white individuals are more likely than whites to have an H. pylori infection and also to develop gastric cancer. And interestingly, the H. pylori positive African Americans are also more likely to be seropositive to gastric cancer associated virulence factors CagA and VacA, and to have a higher antibody level to these proteins. And for us, we still don't know what the underlying reason for this observation is, that the antibody levels are higher among African Americans. This could relate to the host response. So do they have a stronger reaction to the bacterial infection, or is it maybe even the infecting bacterial strain? What we also don't know, and which then may have a public health impact, for example, is whether this is related also to a higher incidence of gastric cancer that is observed among African Americans. And if this would be the case, then this would maybe be an option for precision intervention, because as Meira said, there's antibiotic therapy available for H. pylori, and it is known that this reduces the risk of developing gastric cancer, but antibiotic resistance is developing, so you don't want to just eradicate the bacterium from everybody. You would like to eradicate it only from those that are at high risk of developing cancer. And in addition, some literature suggests that there are beneficial effects of an H. pylori infection, like protective effect for esophageal adenocarcinoma, GERD, atopic diseases, but also obesity. Thank you.
Great, thank you so much for that presentation. We've got a lot of comments in the chat box, which is awesome. To kick us off, I'll go to Jillian Bentley, who has a hand raised. Jillian, you can unmute yourself and go ahead.
Yeah, thank you. So that was a really interesting talk. Thanks very much. And my comments are more from an anthropological perspective, but I think they have strong bearing on what you've been talking about here. And the first of them relates to the question about using the term race, because it implies that there's some kind of genetic difference between African Americans and whites. And, you know, one could go into a long argument about that. But the second is that, you know, there's very clear social disparities between African Americans and whites, and, you know, disparities relating to lack of health care in the USA. And you did control for some socioeconomic variables, but considering there's such a strong environmental component related to H. pylori, I'm just wondering, you know, why you didn't consider things, maybe there's no data about this in your particular data set. They were about hygiene in the early environment for people who are perhaps growing up in poverty, and all of these other areas. So I just wondered if you could comment more about that, you know, because also there are studies out there in relation, for example, to prostate cancer differentials between African Americans and whites. And when you remove some of these socioeconomic factors, you find that there's actually no difference in morbidity and mortality between these different groups, which suggests, you know, that has nothing to do with genes, but it has everything to do with the environment.
Yeah, so sorry we didn't get into this at all. The Southern Community Cohort Study is a really interesting study. As Yulia mentioned, they recruited people from community health centers, so places that serve the poor and underserved, uninsured, back in the early 2000s. And one of the things they did for some of our subset of data, and I have the data too, is they did ancestry informative markers. So the goal, as it was funded by the NIH, the study was to look at racial differences in cancer, taking away the socioeconomic status as much as possible, right? Recruiting people equally. So this is a very low income group altogether. 65% have an annual family household income of less than $15,000 a year. And when we did the ancestry informative markers, so they actually sought to recruit people who said, I am white only or I am Black only. They were trying to look at these groups. And so when they did the ancestry informative markers, they found there was no overlap at all. So the people who self-identified as African American only, the aim said they were 50% to 99% African. I think the average was like 92% African. And among the people I self-identified as white, I think it went up to 15% African. It was on a 0 to 1, white to Black scale, and the average person was a 0.7% African. So it's absolutely self-identified. We also have these ancestry informative markers, so they separate a little bit. So, you know, definitely, you know, in a cohort study, we don't have medical data. We don't have a lot of other questions, certainly about hygiene. I have a published study, I can't remember, 2014, 15. I got neighborhood level SES markers from census block information, and we looked at neighborhood level factors and individual level factors. And one thing really interesting is that the greater percent African ancestry, the greater likelihood of carrying a CagA positive H. pylori. And once you adjust for all of the strains, and that doesn't necessarily follow SES, but once you adjust for all of the neighborhood and individual level factors, it was still something an odds ratio of 9 for a person of in the highest category of African ancestry carrying a CagA positive H. pylori strain. And if we remind you of the enigma in Africa, I still think that H. pylori is one of those pathogens that actually is a commensal that did a lot of good things. So if you have H. pylori, you're less likely to have gastric reflux, right? You have a bacteria that's modulating the gastric acid levels. So it can, you know, create a niche for itself, but also reducing some of the current Western epidemics we're having in acid reflux and GERD. So yeah, all good questions. And I hear you with prostate cancer. I still think because we know about the genetic diversity of the bacteria, I think there's something there within the disparities to the bacteria itself and the host response to the bacteria.
Okay, thanks.
Great. So I think we have a question from Tony Baines next. Tony, you can unmute yourself and go ahead.
Hey, how you doing?
Hey, Tony.
It's good to hear from you. Just a quick question. So, you know, you focus a lot on African Americans, but I'm just curious to know, do you see these same observations with other Black populations, Africans, Cuban, Afro-Americans? I mean, just different groups of Black population. I'm just curious, do you see these same observations, you know, that may be dependent on geographics?
Yeah, it's a really good question. There hasn't been that much work done. I will say we see the same associations with Asian Americans. We just don't have the Asian American data, but you definitely see in Asian Americans this higher antibody response and higher incidence. I can tell you there's some really interesting work out of Colombia. We have the slides, we just didn't have time to show it, where they were looking at, in Colombia, people of African ancestry and people of indigenous ancestry, and their people of African ancestry in Colombia have less gastric cancer. Whereas the mestizos, the people of indigenous and European mixed ancestry have more. And there's a lot of interest in diet. And as we mentioned, that we know that salt is an increased risk factor, as is smoking, and possibly fresh fruits and vegetables decrease risk. And at least in the areas they've been looking at in Colombia, the African Colombians live closer to the water, and we think they have a better diet than the indigenous Europeans who live in the mountains. Less access to fresh fruits and vegetables, more preserving of foods by salt. But I completely hear you. I think that it varies, and we don't have the data, but I know that in Africa, they're starting to see more gastric cancer, unfortunately. So the African enigma might not hold up, and this might be related to increasing Western lifestyle in Africa. But I know that people are starting to look at this more.
Great, thank you. Next, we have a question from Tara Sipon Robbins. Tara, you can go ahead with your question.
All right, hi, thank you. So how early would we expect to see these disparities age-wise? Because we're working in a rural southern population as well, and we found really high rates of fecal calprotectin in, so high intestinal inflammation in kids ages about 3 to 12, and it's higher than any other comparative sample that we're seeing. So I'm wondering, and the nurse has reported really high levels of H. pylori in the community as well. So I'm wondering if there could be something at play with long-term exposure in children, and sort of when exposure occurs and the immune response over an extended period of time, if that could be at play. So I don't know if you can speak to that at all.
The work I've seen, I'll let Yulia jump in if she's seen something else. I saw some really great work out of Texas. I'm so sorry that my dog is barking. But out of Texas, in child care centers that serve low-income kids, they saw a, but they saw, again, SES is strongly associated with H. pylori infection. So they saw overall, a really interesting study they did is that they eradicated the H. pylori from these kids. And what they found is that within 1 year, the white kids had about a 6% reinfection rate, and African-American kids had about a 50% reinfection rate, right? So yeah, I think there are disparities as children. Yulia, do you have anything to add to that?
No, I don't. I'm not, no.
Yeah, no, it's a good question. Again, there hasn't been that much work done on H. pylori in children. I think normally when it's an acute H. pylori infection, they go straight to the GI and it's eradicated. That certainly happens.
It would be really cool to get longitudinal data leading up to then the adulthood stage, and yeah, cool. Thank you.
Absolutely.
Great, thanks. Next, we have a question from Charlie Nunn. Charlie, go ahead with your question.
Sure, I posted some in the chat, but you brought up Marty Blazer, and I'm just interested as well in what you think about some of the proposals he's had that Helicobacter provides benefits early in life, you know, and cost later in life, and sort of an antagonistic pleiotropy kind of framework in the word, you know, in the framework of evolutionary biology. I wonder if you have any thoughts on that, what the evidence is for that kind of trade-off across the life course.
I think that makes a lot of sense. I haven't seen the obesity play out. We wanted to include it there because he was a proponent of that. I think he's not as much a proponent of that anymore. His data on atopic diseases, so allergy and asthma in children, it does look pretty good. It makes a lot of sense. It kind of can go along with the hygiene hypothesis as well. So I think, you know, when Yulia and I are trying to discuss, do we need to screen and treat, we're really thinking of an adult population so that one can still be provided the benefit of H. pylori as long as it didn't become acute with, you know, pain and gastritis. And then at some point, say age 40 or 50, because gastric cancer is traditionally a cancer of older people, that's when we come in and eradicate it. So the question then that everybody wants to know is if you do eradicate it even as an older age, what's happening then? So if you take out an ancient microbe, what microbes take its place? Something that didn't evolve to live in that person's stomach, right? I can say of the half dozen now clinical trials to eradicate H. pylori, there has yet to been a report that after eradicating H. pylori, now everyone's getting esophageal cancer and GERD and asthma and obesity. So it's not necessarily that I think it diminishes the protection you maybe have gotten from harboring H. pylori as a child. And again, I think that could be why we're starting to see greater rates in Africa where they have H. pylori but have other competing organisms. There's some great studies that if you also have helminths and H. pylori, the H. pylori is less likely to lead to gastric distress and cancer. But as we resolve the other infections and as people live longer, they're at greater risk of developing gastric cancer. But I think the protective effect of H. pylori has not been worked out at all, but makes a lot of sense that there's a reason that it stayed with us all of this time.
I was also wondering if you could say something about the evolutionary origins. If I understand from the Moodley paper, you were saying it was introduced by their analyses about 100,000 years ago. And so it's not something that has sort of long-term millions of years of coevolution with humans, at least according to that analysis. Which, you know, your response to the other question just made me think of. I mean, of course, that's still a long time. It could certainly do, you know, create a lot of coevolution or kind of signaling for the immune system, and, you know, things like that for training the immune system in sort of an informal term. But what's the original? Is that the widely accepted interpretation? And what was the source of H. pylori if it didn't co-evolve with humans for so long?
Yeah, I don't know. I have just accepted that, not being in that field, but I think you have really good questions, and I'm not sure who's looking into that. Do we know, Yulia, if there are groups looking into that? Yeah, I don't know.
That's really
project for someone in the audience. So thinking about kind of the protective effects of Helicobacter pylori, there's a question from Melissa Melby in the chat that goes along those veins. Melissa, if you're willing to ask verbally, that'd be great.
Hi, I'm keeping my video off because I have kids wandering around here. Yeah, the last one, thank you, Charlie, for asking my one about Marty Blaser, but I guess I'm still wondering about the antibiotic resistance issue. Is that the one you're talking about, Meredith?
Yes, yes.
Okay. Yeah, just given that you were talking about double and triple antibiotic therapy and thinking about more than H. pylori, but more about the microbial communities in which H. pylori resides, anytime we treat a bacteria like H. pylori with antibiotics, we're generally knocking out quite a lot. And I wonder if we might expand our perspective instead of focusing on a single bacterium and really look at the microbial communities to get a better understanding of some of these population differences. And it also allows for a little more evolutionary approaches there. Can you just comment on that?
Yeah, I think that's really interesting. You know, when we talk about the microbiome, we're usually talking about the intestinal or the gut microbiome. By living in the stomach microbiome is not as large or diverse. Interestingly, studies have shown that when you have H. pylori, it's like 95% of the bacteria in your stomach. When you don't have H. pylori, you have a much more diverse microbiome. So there could be benefits to eradicating the H. pylori because in general, we think diversity in your microbiome in the colon, for example, is good for reduction of colon cancer. We don't know about in the stomach. Yulia and I also do work in H. pylori and colon cancer, actually, and we think there might be some downstream effects of the stomach microbiome to the colon microbiome. So there are all really good questions. I know that there have been studies of actually a number of people, when they give H. pylori antibiotic therapy, they give probiotics at the same time. And it appears that when you give probiotics at the same time, it does not reduce the efficacy of the antibiotics. In fact, it seems to help people sometimes with the side effects of taking antibiotics. How quickly do you get back to a normal microbiome? I don't know. And obviously, we're also concerned about reinfection. But you're absolutely right that H. pylori lives in a broader environment of other microbiota. And certainly, we don't want to be throwing antibiotics and more antibiotics at folks. You know, it's a cancer-causing agent, and this is a very serious cancer with really high fatality. So it's hard to not want to do something about this. I think when I started in this field, and I worked with Marty Blaser back when I was a postdoc a long time ago, he had mentioned once something I love, this idea that could we find out if you are harboring the wrong strain for you, right? Either it's a host bacteria mismatch, or it's a highly carcinogenic strain, we could eradicate that strain and then give you a probiotic of a good H. pylori strain. So return that ancient microbe to you, but in a way that is more harmonious with your environment. I don't think that's ever happened, and nobody's volunteering to be given H. pylori except for Barry Marshall, the Nobel Prize. So yeah, no, those are really good questions, and we're trying to look at that. We did a small study, and I used to be at Vanderbilt in Nashville, where we collected stool from folks who were H. pylori positive and not, and tried to look at is the microbiome in the stool, which is mostly the intestine, not the stomach, varies by H. pylori status, and we actually didn't see it. We saw more differences by race than we saw by H. pylori status. So yeah, it's definitely an area to continue to look into.
Thank you. Can I just ask one follow-up question about that too? I guess my understanding is that sometimes individual organisms, depending on where they reside in the GI tract, can act either as pathogens or as commensals or even very beneficial organisms. And given that we tend to rely on stool samples, is that possibly a a limiting factor in a lot of the analyses that are going on in terms of we don't know where they're residing in the GI system?
Yeah, we have a new study on it. People are doing work on this too, trying to get more biopsies. And it's hard, of course, to scope somebody when they're otherwise healthy. Which is what a lot of, unfortunately, people who have no symptoms to H. pylori still develop gastric cancer because of their H. pylori. But actually, people are actually really looking into this. And actually, I think this is an exciting area of where the H. pylori resides and the variation in that. So that is starting to be more an area of research. Oh, I saw somebody's little quote, which I thought was really interesting. Yeah, we both tested ourselves for H. pylori, and neither Yulia and I were positive. And I don't want to say I'm disappointed, but I was. But yeah, no, H. pylori somehow. And Ashkenazi Jews, we have more H. pylori too. Really.
Very interesting. So I'm curious about kind of the serology in the paper. Does that detect different strains of H. pylori, or is it just a general response to H. pylori? And I guess how much information do we have about the different strains and whether there's co-infection of different strains, things along those lines?
Yeah, so it's so all proteins that we have in the array all come from one strain, right? But as I said, there are some strains that don't even have the CagA gene. So if we measure antibodies to CagA, they most likely come from a strain that has CagA, right? So, but it doesn't necessarily mean that if we don't measure antibodies to CagA, that the person doesn't have a strain that has CagA. Because it's only, antibodies are always only an indirect measure. We never detect the strain itself. So that's a drawback. And I wouldn't say that it's the perfect assay to determine the type of strain that a person has, right? But it's, well, close enough, I would say, to maybe identify markers that are associated with an increased risk of gastric cancer, for example. And it is much more feasible as an assay than scoping people and extracting strains, for example, and do sequencing or anything. So yeah, it has advantages and disadvantages, let's say it like this. So, but we can't say for sure that this is a certain strain just by measuring the antibody levels.
Great, thanks for clarifying about that. So there's been a lot of discussion about kind of the racial aspects of the study. And there's a question about using the dichotomy of African Americans versus white when there's so much introgression and genetic diversity. Is there any genetic basis for using the African American versus white dichotomy? I know it was self-reporting, so it was a little limited, but just wondering if you could speak a little more to that.
Yeah, I can share the screen too. So I mentioned before, we did do ancestry informative markers, and we have done the analyses purely by the ancestry informative markers. Again, in this study, they were trying to recruit people who self-identified as white only or black only, and they really, by the ancestry informative markers, are completely, they didn't get admixture. This is what they were looking for the most part. But if you allow me to share my screen one more time, I will show you some, oh, there we go. Some really quick data by ancestry. So here are our ancestry informative genetic markers, and the dark blue is the white, and then the African Americans are in the three groupings, and low is 50 to 85% African ancestry according to these AIMs, these AIMs. And again, there's no crossover with the whites because the highest was 15%. And you can see for H. pylori, but especially for those CagA antibodies, that it really separates out, surprisingly, by ancestry informative markers. So I absolutely appreciate that race is a social construct as well as a biological construct, if you believe that it's a biological construct as well, and that it's combined. I will say in this study, this is the data that we have, so that they are separate. There were some other really great questions I saw in the chat box about cofactors that are not biological, right? So stress and discrimination, and how that might affect the immune response and why we might be seeing thus these higher antibody levels. So again, we absolutely agree. It may not be that they have a more carcinogenic strain. It may be the host response to it in addition to all the other factors. This is something we are always exploring and interested in. And I agree that we can't control for all of it because we don't have the data on all of it. But I think that's a really important and interesting thing to see and to think about. I know that higher antibody response to an infection is not special to H. pylori, that people see it in other infections as well.
Thank you for speaking more to that. So we probably have time for a couple more questions. I know there's a lot of questions in the chat that we have not gotten to. So if you raise your hand, that'll be the quickest way to get your question addressed. Looks like we do have a hand raised from Margaret Humphreys. Margaret, you can go ahead with your question.
Thank you. I'm curious if there are any differences in diet that you can point to, that a high carb diet, a high fat diet, a high specifically glucose diet or sucrose diet makes a difference in the, does the H. pylori thrive better in different food created stomach environments, I guess would be a way to put the question.
Yeah, it's a great question. There's lots of interesting studies on nutrition and H. pylori. We did some work again in the Southern Community Cohort study looking at circulating nutrient levels and cancer prevention research. I don't remember when, 2015 maybe. Anyways, we saw that among people with H. pylori, but especially among people with CagA positive H. pylori, they have these lower circulating micronutrient levels and like beta carotene. And so there's a lot of questions about if you have H. pylori, it's actually harder to uptake nutrients. H. pylori needs iron, so it takes iron. So the iron deficiency anemia is also associated with H. pylori. Versus if you have H. pylori, are you eating differently, right? Based on how you're feeling in the stomach and how, so H. pylori, a lot of people with H. pylori actually are sometimes lower weight, and then once their H. pylori is eradicated, they might gain a little bit of weight, I think often because they're feeling better. So how the bacteria affects what you eat affects the bacteria are really good questions. There was a really neat study in India in a very malnourished population where there was a lot of H. pylori. And before giving them, golden enhanced rice, they actually eradicated the H. pylori first and then gave them the enhanced foods, and then it seemed to be greater uptake of the vitamins. So those are all good questions. In epidemiology, it's hard to see. We do sometimes see a reduced risk with fresh fruits and vegetables, and we see again that salt, especially salt preserved red meats, hot dogs, ham, sausages, those all seem to be synergistic with H. pylori, unfortunately, and to increase the risk of gastric cancer as well.
Thank you.
Great, thanks so much for that. So I'm curious about a question that Charlie posed at the very beginning in the chat, whether there are any hypotheses for what factors drive the variation in virulence or carcinogenicity among the different strains of H. pylori.
Yeah, you have an answer for that.
No, I saw that question and I unfortunately don't have an answer to that because as far as I remember from my evolutionary classes, it doesn't make sense for the bacterium to kill the host, right? So I don't. So I don't know why these carcinogenic strains specifically evolved from the others. So the CagA gene is on a pathogenicity island. So this might have happened through gene transfer between bacteria, maybe from a different bacterium. And maybe these are especially or specifically able to survive in the gastric niche. But this would be my only theory, that they receive some benefit from getting this gene, and then better surviving in the stomach. But otherwise, I have no idea. Yeah, I'm not aware of any studies to this either. So.
Yeah, it'd be super interesting to know kind of what additional things they're getting by being more virulent. But yeah, thank you for that response.
Yeah, somebody posted, Sandra Broom Anderson posted, production of some of the virulence factors is related to nutrient uptake by the bacteria. And I guess as well, well, at least in the case of cancer, that happens relatively late in life. And so maybe there's not really a lot of, selection against that higher virulence, at least in the case of cancer, because it happens in the selection shadow.
That was my thought as well, is that there wouldn't be as much host pressure against selection for virulence, but it would be driven by selection for virulence on the pathogen side.
Right. Anyhow, we're almost at the top of the hour, and so we need to close things down. I feel like we could go on for another hour with all of the really fantastic questions that are still popping up in the chat box. This will all be made available, you know, on the recording, on the record of this, and so people can, you know, look through these and follow up with one another to talk about some of the questions further, and we really hope that that happens. This is just such a great topic. When I teach evolutionary medicine, I always cover H. pylori in multiple places in my syllabus because it highlights so many of the core concepts from evolutionary medicine, and it's just a great example and so provocative, you know, for the students and for everyone else. So thank you both very much for a fantastic presentation, for taking the time in your busy schedules to participate. I just want to, and I want to thank everyone else as well for joining us. I know a lot of us are juggling kids and teaching started up and everything else. So thank you all very much for coming. Thank you, Meredith, for organizing everything, keeping it running so smoothly. I really appreciate that. I just want to call your attention to the next event. We have Amy Boddy, who's an assistant professor of anthropology at University of California, Santa Barbara. And she's going to be talking about cancer prevalence and life history traits in mammals. And this is really an opportunity to start highlighting some of the articles that have been published in Evolution Medicine and Public Health, the journal that's part of the society, and I'm the editor of that journal. And so we're really excited to have Dr. Boddy present her work, a paper she recently published. We'll share that with everyone. If it hasn't been shared already, maybe it has. And we're also going to hear from a couple of probably her co-authors, I understand are going to join, but also a couple of other experts who are going to weigh in on some additional perspectives that have emerged from this work. It's really created some other kind of discussions and perspectives that I think have been really valuable and a really good discussion that we've been having in the journal, and we just want to share it with the audience here in Club EvMed. So with that, I think we'll close things down. Anything else, Meredith, that we need to say?
Nope, that's everything. Thanks for coming, and I'll send out a recording of this meeting to everyone soon.
Thanks, everyone. This is great.
Thank you.